biodiversity, they are of great importance not only to animals but also to humans. However, because of ocean acidification, bleaching as a result from increased sea surface temperatures, overfishing, and eutrophication, coastal areas are being threatened. It's predicted that due to global warming, the sea surface temperature will increase with up to 4 degrees Celsius by 2100, leading to irreversible damage to coastal areas and reef degradation. In addition to this, eutrophication can lead to oxygen deficiencies and algal blooms. This might also lead to a regime shift from corals and crustless coralline algae to macroalgae, sponges, or benthic cyanobacterial mats. Due to the variability in coral response to acidification and global warming, it is likely that reef degradation will occur at a greater temporal and spatial heterogeneity than previously expected. On the other hand, it's unlikely that coral reefs will disappear altogether, but rather they will change, as some species have been showing an increased resilience to climate change. However, there remains much unknown about coral reefs and coastal ecosystems, especially regarding the effect of different future climate scenarios. For this purpose, marine lakes are an ideal model system. Marine lakes are landlocked and kyaline basins, meaning they are subterraneously connected to the ocean via tunnels, cracks, and fissures, allowing for some tidal influx. Their characteristics vary depending on their degree of connectivity, with the more connected lakes resembling a lagoon in water chemistry and biota, while the more isolated lakes contain brackish water and unique species. The level of connectivity is determined by the extent of the tidal amplitude compared to that of the ocean. And lakes with a high level of connectivity tend to be holomictic, as they are well mixed due to the stronger influx from the ocean. Isolated lakes, on the other hand, are typically meromictic, as they are well mixed due to the stronger influx from, meaning they display vertical stratification in regard to, amongst others, oxygen gradient, pH, temperature, salinity, and nutrients. Additionally, the carbon and nitrogen source differ as well, depending on the level of connectivity. Coastal ecosystems are coupled to land-derived nutrient sources, for example, by sediment runoff and forest litter, and this influx is amplified by the constant rainfall in tropical areas. However, this effect is overshadowed by the marine influx in lakes with a higher connectivity, where the main carbon source originates from marine organisms, like phytoplankton and marine plants. Marine lakes are a very suitable model system to study, for example, the functionality of food webs in coastal ecosystems, as they function as mesocosms. And the mesocosms are small, closed, simple ecosystems that change little over time, permitting for replicate field measurements of biological and physical attributes. Additionally, they resemble a potential coastal area in the future, as they already have a high temperature and a low pH. Furthermore, as these environmental factors vary with the level of connectivity, the less connected lakes show a higher impact of climate change and eutrophication compared to lakes with a higher connectivity. Thus, marine lakes allow to study future climate change scenarios and to predict the responses of coastal species communities to different abiotic factors. Dozens of these tropical sea-level marine lakes are located in the limestone island of Misol, part of the Raja Ampat Isles in West Papua. In these lakes, Brachydontis mussels are one of the few genera always present, and thus they make for a suitable proxy to use as a reference point to compare the food webs of the different lakes. To determine the food web structure of an individual lake, however, information about the carbon source and trophic levels of biota is required. It's possible to assess the origin and cycling of carbon and nitrogen in a food web by performing a stable isotope analysis. This technique makes use of the delta C13 and delta N15 stable isotope concentrations in organisms to track the carbon source and trophic positions in a food web. This is based on the principle that the delta C13 concentration is carried over to a higher trophic level in the food web. Moreover, the main source of the carbon influx differs between lakes with a low and high connectivity, and thus these sources have a different delta C13 concentration. with the run of a forest litter having a considerably low concentration than the marine organisms like microalgae, as you can see in the table. Additionally, by determining the delta N15 concentration, it is possible to determine the trophic position of an organism in a food web, as this isotope accumulates in higher concentrations when the organism belongs to a higher trophic level. So, if the delta C13 and delta N15 stable isotope concentrations of organisms are known, it is possible to construct a food web of a specific marine lake. And furthermore, if the food webs, these food webs of the different lakes can be compared to each other by using a common factor, the presence of brachydontis mussels, as a reference point. So the goal of this research is to determine the carbon source and trophic positions in food webs from 6 different marine lakes in Raja Ampat, based on their connectivity to the sea. As of yet, relatively little is known about the food web composition of marine lakes, especially regarding how this is influenced by the level of connectivity, and additionally, climate change and eutrophication are threatening coastal ecosystems, and marine lakes could be a proxy for future coastal areas, and therefore provide a relevant source of information about potential food web functionality under different environmental factors. Thus, this research could enable follow-up studies to better predict future coastal ecosystem scenarios and subsequently aid in the preservation of tropical marine areas. I aim to answer the following question: how does food web complexity of marine lakes shift with a different degree of connectivity to the sea? And this question will be answered by first determining the delta C13 and delta N15 concentrations per organism present in the lakes. And Ikbar will tell you more about this process in a minute. Next, the food webs of each individual lake will be constructed with the use of these stable isotope concentrations. Furthermore, the roles of the different functional groups will be able to be derived from this. And finally, the effect of connectivity and subsequently temperature on the food web structure can be determined by comparing the food webs of the different marine lakes with each other. The hypothesis is that complexity of food web structure will increase with a higher level of connectivity to the sea. So in lakes with a higher connectivity, more trophic levels are expected to be present, which can be detected by a broader range of delta N15 concentrations, as this isotope accumulates in higher trophic levels. Additionally, the carbon source is expected to have a mainly marine origin in more connected lakes, and therefore a higher delta C13 concentration will likely be found in lakes with a higher level of connectivity compared to lakes with a low connectivity that have a mainly terrestrial carbon source. And now Ikbar will continue with his research question.
Thank you, ladies and gentlemen. Okay. As my partner Martijn has explained before, that we are working here together within the same project, also in the same process and the same data and the same analysis. But then I take focus on one specific species called Brachydontes. And this is my research question, ladies and gentlemen. How does Brachydontes differ in regards of spatial and temporal point of view? In this research question, I want to highlight the keyword. The keywords are Brachydontes, spatial and temporal point of view. So I want to know how the difference on Brachydontes among the 6 different lakes. In total, we are going to do analysis on 6 different lakes. I want to know the difference. After that, I want to also know how Brachydontes differ if we want to draw the time back. At least we have the data from now 2020, and then if we move back, we also have the data from 2017. So with this data of delta carbon 13 and delta nitrogen 15 of Brachydontes, we can draw and we can depict the difference of Brachydontes due to the time and due to the space. Okay, then what are the hypotheses of the research? At least I have 2 hypotheses. First hypothesis is that in the less connected lake, which is more isolated lake, the value of carbon, the delta carbon 13 will be lower because the main input for this lake, this isolated lake, is coming from the terrestrial area. Meanwhile, the highly connected, the more not isolated lake, they have input from surrounding ecosystem, especially from the oceans, like that. And the second hypothesis is related to the berjudul test itself. The berjudul test is a filter feeder animal, so it uptake the water and then take the nutrient and also the other organism. And due to this species can be used as a proxy or the baseline because it is also situated in the low level of the trophic level. So we assume that if there is a shift on this berjudul test, the species or organism which are located above the berjudul test will be also shifted into the other way. Ladies and gentlemen, this research is conducted in Papua, in Misol, West Papua, Raja Ampat, Indonesia, and we are very proud to say that in total we are working with more than 2,000 samples. Once again, more than 2,000 samples, and this is very challenging project. Many people said this is a quite simple project, but the challenge is real. I mean, for 2,000 sample. Previously, we are working with 11 lakes. Unfortunately, due to this corona outbreak, we are only able to finish 6 lakes, but at least we put them in the right category, represent low connectivity, medium connectivity, and high connectivity. We took 3 pickets per species. After that, we grind the samples and then analyze using the stable isotope analysis, and then do statistical analysis here. Okay. In general, I want to show you the research flow that we have been collecting the sample. At least Pak Ludi, Mr. Ludi, Lisa, and the teams, they went to Papua, I think last year in 2019, to collect the samples. And currently we have done grinding the samples, more than 2,000 samples, and we are moving towards to decalcification process to decalcify the calcium carbonate of the sample. For example, a shellfish, that it needs to be decalcified to eliminate the calcium carbonate. And then we go to the sample weighing. After that, we transport the sample to NIOS to do the stable isotope analysis. The data that we have obtained from NIOS, we have the delta carbon 13 and delta nitrogen 15. We are going to analyze it using stable isotope analysis. First, we have CR, stands for stable isotope analysis in order to determine the food proportions or diet proportions. For example, berjudul test has how many percent of this food source, something like that. And then we are going to also work on SIBE, stands for stable isotope Bayesian ellipse in order to determine the niche space width of every sample species. In general, this is called stable isotope analysis. We are working on carbon 13 and nitrogen 15. Why? This is a heavy isotope, right? In general, we have 2 light and heavy isotope. The difference is only the number of nucleus. This is obtained from Book of Fry in 2006. Let's say the heavy isotope, it has the stronger bonding and not really fast in reaction, so it is retained in the tissue of organism. It is quite stable compared to the light isotope. The carbon, it is predominantly coming from the source of the food, and the nitrogen, it is enriched due to the assimilation process of the tissue of the animal. How to look at this? It is simply by looking at the comparisons of the sample and the standard. For the carbon, we refer to the limestone, and the nitrogen, we refer to the nitrogen in the atmosphere, and then multiply by the 1,000. So this is the planning and paid up. We expect to finish it in the end of February or the early of March. However, if something that happen, something bad happen due to the coronavirus, for example, there will be a total lockdown, we will have to do the backup plans that we are going to analyze the existing data from the previous year. So we are not able to do lab work anymore. So the solution is working on the previous data. And thank you. This is from us, our supervisor. The first supervisor is Mr. Ludi and Ms. Didemas and me, Ikbar and Martijn. Thank you for listening. And I will give the stage back to Martijn.
Nice.
How is it? It went well?
Yeah, I think so. And in total, we took about 18 minutes.
18 minutes. Cool.
Yeah, we have 20, so that's fine.
Cool. Yeah. And I also record this already.
Oh, yeah, I see. Okay.
So I could